Hello students, welcome to zoology class. Today in continuation to my presentations on cell cycle and cell division, this is the second presentation. In my first presentation I talked about the cell cycle, and now in second presentation I will talk about the cell division. And the cell division, it is of two types. One is the mitosis and the second one is the meiosis. And in this present video presentation I will talk about the mitosis. So what is mitosis? Actually, mitosis occurs in somatic cells. It means all the cells other than sex cells, and it is known as the equational division. So mitosis is a type of cell division which occurs in somatic cells, and it is known as equational division also because the chromosome number in the parent cell and the daughter cells is the same. And mitosis, it is the phase of the cell cycle where the nucleus of the cell is divided into two nuclei with equal amount of genetic material in both the daughter nuclei. It succeeds the G2 phase of the cell cycle and is succeeded by cytoplasmic division after the separation of the nucleus. It means that after the G2 phase of the cell cycle, mitotic division starts, or the mitosis starts, and in the mitosis the nucleus of the parent cell divides into two nuclei, and after the division of the parent nucleus in two nuclei, then it is followed by cytoplasmic division. Then the cytoplasm of the parent cell also divides into two, that is known as cytokinesis, and then two complete daughter cells are formed as a result of the mitosis. And mitosis is essential for the growth of the cells and the replacement of the damaged or the worn out cells. And abnormalities during mitosis can change the DNA, and which results in genetic disorder. So this is mitosis or the equational division, and these are the features of mitosis, that in each cycle of cell division two daughter cells are formed from one parent cell. And as I already told you, that this division is also known as equational cell division because the chromosome number in the parent cell as well in the daughter cells is the same. And in plants, mitosis or the mitotic division, it leads to the growth of the vegetative parts of the plants like root tip and stem tip, etc. So these are the features of the mitosis, and you should know that mitosis is the division of the cell, which is also known as equational division. It occurs in somatic cells, and chromosome number is same in the parent cell as well as in the daughter cells. And as a result of one mitotic division, two daughter cells are formed from one cell. So this is basic of the mitosis. So what are the stages of the mitosis? So right before the first stage of the mitosis, that is the prophase, the cell spends most of the life in the interphase stage of the cell cycle, where preparations are made before the beginning of the mitosis. So DNA is doubled, all the material is doubled in the interphase stage, and after the preparation for the cell division, the mitosis starts. And since the actual process involves the division of the nucleus, prophase is technically the first stage of the process, because in prophase division starts, and the different stages of mitosis occurring during cell division can be studied as under. So first of all, there is interphase stage, which is the preparation for the mitosis, and in interphase stage cell grows in size, and the cell spends a period of its growth under interphase, and in this interphase stage there is G1 phase, which is the period before the synthesis of DNA, and S phase, which is the stage of the DNA synthesis, then G2 phase, which is the phase between the DNA synthesis and the beginning of the first stage of the mitosis, and that is the prophase. I have already taught you about the different stages of the interphase, and then comes the mitosis, that is the actual division of the cell. So first stage is the prophase stage of the mitotic phase. Mitotic phase has the first phase that is known as the prophase. So prophase immediately follows the synthesis phase and G2 phase of the cell cycle, and it is marked by the condensation of the genetic material to form compact mitotic chromosomes, which are composed of two chromatids, and these two chromatids, they are attached at the centromere. And the completion of the prophase, it is characterized by the initiation of the assembly of the mitotic spindle fibers, microtubules, and the proteinaceous components of cytoplasm that help in the process of division, and the nuclear membrane, it starts disintegrating. So this is the prophase cell. So this is the nuclear membrane which has started disintegrating, and these are the spindle fibers which have started forming, and these centrioles, they have moved to different portions, and there is condensation of DNA. Chromosomes appear as chromatids. They are joined by centromere. Nuclear membrane tends to disintegrate. Nucleoli, they starts disappearing, and in animal cells, centrioles, they move to opposite poles like this. One centriole is here and the other centriole is here. So cells at the end of the prophase do not show Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear membrane. So this is the prophase stage of the mitosis. Then the second phase is actually metaphase, but before the metaphase we can discuss prometaphase. And in the prometaphase stage of the mitosis, the nuclear envelope disintegrates completely, and microtubules are allowed to extend from the centromere to the chromosome, and the microtubules attach to the kinetochores, which allow the cells to move the chromosomes around. So these are the spindle fibers, and these are attached at one end with the centriole and with the other end with the centromere of the chromosome. So then comes the metaphase stage, and in metaphase stage there is complete disintegration of nuclear membrane, and microtubules, they start pulling the chromosomes with equal force. So these are the microtubules, are the spindle fibers which pull the chromosomes with equal phase on this side as well as on this side, and this region is known as the metaphase plate. So here where there is assembly of the chromosomes, this forms the metaphase plate, and each cell gets an entire functioning genome, and the chromosomes, they are spread in the cytoplasm of the cell, and spindle fibers are completely formed in metaphase stage. And the metaphase plate is also known as equatorial plate or the metaphase plate, and nuclear membrane is absent, nuclei are absent, centrioles are at the opposite. So this is the metaphase stage of the mitosis. Then next stage is the anaphase stage. So this is a very rapid phase. This is the third phase actually of the mitosis. This is a very rapid, fast stage, and in this stage the splitting of the sister chromatids marks the onset of the anaphase. So the two chromatids of the chromosomes, they are split now, and these sister chromatids, they become the chromosomes of the daughter nuclei. So half the chromosome goes to this side and half of the each of the chromosome goes to this side. That is, one chromatid goes to this side and one chromatid goes this side, and this one chromatid, this forms the whole of the chromosome in the daughter cells, and the chromosomes, they are pulled towards the pole by these fibers, and the centromere of each chromosome leads at the edge while the arms trail behind it. First the centromere goes and the arms goes behind it, and the separated chromatids, they are now called as the chromosomes, and they start moving towards their opposite poles, and by the late anaphase each pole has one set of chromatids, and each chromatid, it gets a centromere, and then it becomes a complete independent chromosome. So this is the anaphase. Next comes the telophase. So in telophase, this is the final stage of the mitotic phase, and in this telophase the chromosomes that cluster at the two poles, they start collecting at the poles into an undifferentiated mass, and as the nuclear envelope, it starts forming around it again, and the nucleolus, Golgi bodies, endoplasmic reticulum, which had disappeared earlier, they they start appearing again after the prophase starts to reappear, and telophase is complete, and now there are two groups of the chromosomes, and this telophase stage, it is followed by the cytokinesis. Cytokinesis, it is actually the division of the cytoplasm after the division of the nucleus, and it is the division of the cytoplasm to form two complete cells, and it marks the completion of the cell division. And telophase, it is followed by cytokinesis, and it is the division of the cytoplasm to form two daughter cells. So this is the completion of the cell division. So these are the four stages of the mitosis, that is prophase, followed by metaphase, then anaphase, and then telophase, and telophase is followed by the cytokinesis, which is the division of the cytoplasm to form two complete daughter cells. So this is the diagrammatic representation of whole of the process of the mitosis. So this is interphase stage, and this is prophase stage. So in this prophase stage, the nucleus is doubled, and here this is a metaphase plate, and this is metaphase stage, and here this is anaphase, and the chromatids, they start moving toward the opposite poles, and each chromatid, it forms a complete chromosome, and then there is the cytokinesis, that is the division of the cytoplasm, and now these two complete daughter cells are formed from one parent cell. So this is the whole of the process of the mitotic division, which is the equational division. So at the starting, the parent cell had two chromosomes, and these two daughter cells, they are also having two chromosomes. So the chromosome number remains the same. So next comes the functions of mitosis. So what are the functions of mitosis? There is a role of mitosis. So mitosis helps in the development of the organism. In single-celled organisms, mitosis is the process of asexual reproduction also, because from one cell, two cells are formed, and mitosis helps in the replacement of the damaged tissues, and the cells near the damaged cell, they begin mitosis when they do not sense the neighboring cells, and the dividing cells reach each other and they cover the damaged cells and new cells are formed as a result of the mitosis. So this is the function or the role of the mitosis. So next comes the significance of the mitosis. That what is the importance? Why the process of mitosis is important. So mitosis is important because it is responsible for the development of the zygote into the adult. Then equal distribution of chromosomes happens to each daughter cells in mitosis. It is also responsible for the growth and development of the individual, and it maintains the constant number of chromosomes in the daughter cells. And mitosis is required for asexual reproduction, vegetative propagation in plant, and is also responsible for repair and regeneration of the damaged tissues. Then mitosis also helps in maintaining purity of the genome. So the purity of genome is maintained because there is no recombination, no crossing over takes place in the process of mitosis, and it is also responsible for repair and regeneration of old and damaged cells in animals and other organisms. So this is the significance of the mitosis. And mitotic division in meristematic tissues in plants results in continuous growth of the plant. So mitosis is very much significant in plants also. So this is all about the mitosis. So these are the four stages of the mitosis. Then there is the role of mitosis and the significance of the mitosis. So this is all about the mitotic cell division, which includes four stages: prophase, metaphase, anaphase and telophase. And this is very much important for the organisms. It is also known as equational division, and it maintains the uh, equal number of the chromosomes in daughter cells. So this is all about the mitosis. I have sent you study material also side by side. You have to prepare notes by combining the material of this presentation as well as the study material, and you have to learn this topic. And if you find any difficulty, you can note down the doubts, and when you come for virtual class, you can discuss those doubts with me. Okay, thank you.